Participants will be on a listen-only mode until the question-and-answer session of today's call. At that time, you can press star 1 to ask a question from the phone lines. I'd also like to inform parties that the call is being recorded. If you have any objections, you may disconnect at this time. And then I'll turn the meeting over to Mr. Paul Mead. Thank you, sir. You may begin.
Great. Thank you very much. Good morning, everyone, and welcome to today's HHS Working Group on Lyme and Other Tick-Borne Diseases webinar on tick-borne disease diagnostics. My name is Paul Mead. I'm the acting branch chief here in the Bacterial Diseases branch at CDC, and I will be the moderator for today's webinar. As I think everybody on the call is probably well aware, tick-borne diseases are a large and growing public health problem in the United States. There are an estimated 300,000 Lyme disease cases diagnosed annually in the U.S., and the geographic distribution of the disease is expanding. There are also less common but potentially serious infections that include anaplasmosis, babesiosis, ehrlichiosis, spotted fever, group rickettsiosis, and Powassan virus disease. And in addition, there are several new agents that have been described recently, such as Borrelia mayonnae and Heartland virus. There are many important topics to cover on the issue of tick-borne disease, but today we will be focusing specifically on recent developments in the area of diagnostics. Today's speakers are shown here. They are Dr. Marty Schrieffer of CDC, who will be speaking on the future of Lyme disease diagnostics, particularly serology. Dr. Claudia Molins, also from CDC, who will discuss the possible role of metabolomics in Lyme disease testing. Dr. Cecilia Cato from CDC in Atlanta, who will be giving an update on rickettsial disease diagnostics. Dr. Malia Elias from NIH, who will be providing some program updates on NIH. And then finally, Dr. Noel Gerald from FDA, who will discuss the role of FDA and provide a summary of the talks. I should note that the views expressed in this webinar are those of the participants and do not reflect the official policy or position of the CDC, the Department of Health and Human Services, or the U.S. government. We will have a single question and answer period at the end of the presentation. Please submit your questions online through the webinar, and we will try to get to as many of them as we can. Please also provide your identity and indicate the speaker to whom you would like the question addressed. Finally, I should note that if there are particular technical issues, I believe you can raise your hand and someone will get online and try and help you with that. So without further ado, let me pass you on to Dr. Marty Schrieffer, who will be giving the first presentation.
Thank you, Dr. Mead, and everyone else who's joined in today's discussion. As mentioned, I'll be talking about Lyme disease diagnostics, focusing particularly on serology, but giving a brief overview on a number of things that have been happening in recent years and possible directions that we may be going. In view of the short time allotted, I'll touch on a number of these things rather cursorily. I'll provide references for further information where I can, and hopefully I'll be able to answer some questions here today or by later communications. We are often asked, particularly in recent years, is serology all we have? What is its performance and utility? And what will Lyme disease diagnostics look like in the coming years? The short answer to the first question is no. There are a variety of tools that are available. Serology is the currently recommended and most widely used, over 3 million tests per year, serodiagnostic tests performed. And as I mentioned, it's the current recommendation still. The performance of serology, which I will refer to as standardized two-tiered serology, has been widely published, but it's often misunderstood and sometimes misquoted. We'll spend some time on that performance today, and if it's properly utilized, it can be very useful, but as I mentioned, it has to be understood. There are limitations, and there are areas that we are working to improve. I'll spend a little time on the third question. As Dr. Mead mentioned, that will be my opinion and not necessarily the opinion of CDC. Moving on, just a brief outline of what I'd like to cover today, the basis of the current recommendations, the performance of standardized two-tier serology, some recent developments in serology which are referred to as modified two-tiered serology, a number of adjunct tests beyond serology. I'll briefly mention a few of those, and some recent developments and the likely directions in the future. Perhaps the biggest challenge to Lyme disease diagnostics are listed here. There are few pathognomonic features of Lyme disease, and that raises the need for laboratory testing, for laboratory confirmation. The challenge in that part of the diagnosis is a sparsity of spirochetes in any clinical sample. So that, for the laboratory end, that leaves us or necessitates the amplifications of markers that we believe are specific, either organismally specific or host response specific to the infection. And these amplifications could be natural by the pathogen itself or the host, and the amplification in the laboratory could be something like culture or PCR. So in the next few minutes, I'd like to go over a couple of these amplification tools. Some of them have been around for many years, although they've been improved over that time frame, and some of them are fairly new. And then I'll focus on serology, a few items listed there that we'll specifically address in serology. And then we'll hear later on from FDA, Dr. Gerald. And I'll just briefly mention a few tests that we believe have potential to improve diagnostics in the future. I want to focus first on, I'm sorry, let me back up just a second. Although we're going to focus on serology, I want to briefly mention a few of the tools that have been around for a while, particularly culture and microscopy, PCR, and we'll look briefly at performance data for those various tools. All of them are in one way or another amplification type tools, but I've frequently in considering where we want to go and how do we get there, I'm reminded of a couple lines from Alice's Wonderland, where the cat asks, please tell me which way I should go, or where Alice asks which way I should go, and the cat says that depends a good deal on where you want to go. The goals, which I think we can agree on, are the best diagnostics for case identification and management, and these will lead to, at least in part, achieving the best public health and disease prevention. And I mention that just in part that there's no way that I believe, or that we believe, that diagnostics is the answer to the disease, but it is a good part of prevention and minimization of serious consequences from this disease. So I've called these tools various roads, and there's a continuum, I believe, of what we've been looking at over the past years and how we hope to move forward. A number of these tools have been around for quite a while, although they've developed and improved. And any tool, be it past or present, needs to be thoroughly vetted and compared with existing tools, for example, compared with serology. Wherever possible, it's important that common use samples be used in these performance analyses. We have a panel of serum samples here at CDC that we've made available to hundreds of laboratories over the last couple of years that enables this for serology, and there are other groups that are trying to collect samples for similar things beyond serum samples. In the end, the approach that we use has to be practical, but it's good to remember that what was practical or impractical a number of years ago may have changed and may now be more practical than it used to be. As I mentioned, I want to spend just a few minutes on two commonly known amplification tools, and those are culture and serology. Shown in this slide are performance results for culture and serology in both early disease and late disease. Early disease marked by erythema migrans in both skin and blood samples, and late disease marked by CSF and synovial fluid samples. As we can see, both culture and PCR have similar sensitivities in early disease erythema migrans patients, roughly 50% or perhaps slightly greater. And in blood, often culture will have an improved performance there over PCR because over time, even a very small number of organisms are able to replicate and be detected. Contrast this with later disease, particularly cases of neuroborreliosis or arthritis with joint involvement. PCR has shown over the years, and more recently as well, that it has applications or utility in later stages of disease. And the information shown in this table was presented a number of years, but there are a number of more recent PCR and culture publications that are listed at the bottom of this slide. In the late 80s and early 90s, serology was a wide open field, and because of the tremendous protocol, performance, reproducibility issues surrounding serology, there was a need to and an accomplishment of standardization in the mid-90s, which is depicted here in this slide. And I won't go into it in any detail. I think most people are familiar with it, but it's basically a two-tiered test in which the second tier is Western immunoblotting, and that was recognized as the best way, at least at that time, to achieve specificity. As I mentioned earlier, and Dr. Mead alluded to, there's a tremendous number of cases and testing that occur. Specificity when 3 million tests are run, and we think that the number of cases is probably about 10, true cases, about 10% of that. Specificity is an important factor to maintain in diagnostic testing. So again, that was, those recommendations were put out in 1995 and are still in place. This slide is really just put up for reference in case anyone is interested in finding out what serologic tests have been reviewed and cleared by FDA through the 510K process. The link is at the top of the page, and if you enter in the product code box LSR, you can find all the tests that have been approved over the last years, and really a goodly amount of data and review information associated with those tests. So I'll move from here specifically into serology, standard two-tier test performance. We can see in this slide that this study has presented two-tiered test performance on all three stages of Lyme disease. Stage 1 often indicated by the presence or often accompanied by the presence of erythema migrans, stage 2 and stage 3, and down the first column on the left, various components of first or second tier testing. For the sake of time, I want to focus on two-tiered testing results, so those are indicated at the bottom of the slide there. And again, looking at STTT performance in early stage 1 and 2 disease as well as late disease, we can see a steady increase in detection. It's quite low in the early stages. I think this is a point at which there's a lot of confusion and a misrepresentation on the performance of serology. Admittedly, it's very low in this early stage, as we would expect for an adaptive immune response, but it increases steadily through convalescence in stage 1, and stage 2 is in the 60% range, late stage disease, quite sensitive. And what is shown here is really the compilation of over 30 studies published a few years ago for this data. And these performance metrics have been reproduced a number of times, and there are a few references there on the bottom right that one can go to see this reproducibility. If we look at this performance graphically, we can see that in the next slide. Again, just reiterating a few of the highlights. Disease or detection in early disease in the first few days of illness after a tick bite is quite low, generally in the below 40% range. Within a week or two, it improves steadily and continues to increase through stage 2 and stage 3. So a couple highlights from this slide. Performance in early disease is low, obviously an area of need. Another observation in both stage 1 and 2 of disease is that there's a wide range of performance characteristics for standard two-tiered testing. And I'd like to spend just a moment focusing on both of those issues. We'll do this early disease and some recent tests that have been published on to address early disease. And we'll also look at variation in early stage disease and try to get some insight into what's going on there. There were a number of studies done in the early 2000s. One of them is depicted here, in which we have EM patients, early disease patients, that are characterized as either localized disease, as indicated by a single erythema migrans lesion, or multiple lesions, indicating dissemination in this case. And these patients were sampled when they presented, and then broken into different categories based on the duration of illness, either 1 week or less, 1 to 2 weeks, 3 to 4 weeks, and greater than 4 weeks. If you look at the standard two-tier performance, all the other data is here, but again, for sake of time, I want to focus on standard two-tier performance. I think it's very insightful to recognize the difference between two-tiered performance, serology performance, in localized disease versus disseminated disease. If we look at just the patients at the 1 week time point, those with localized disease are only detected at a rate of about 9%, whereas you look at persons with disseminated disease, dramatically different, 65%. And this trend continues out for several weeks. It's not really until the 3rd or 4th week that persons with localized disease catch up in their humoral response and detection by standard two-tiered testing to those with disseminated disease. And I think this is a factor that affects performance of or detection in all stages of disease. Those that are localized, even going into stage 2 disease, and of shorter duration, are going to have a lower detection rate than those with longer duration. The summary of that table is depicted here, and again, just for graphic purposes, showing the difference between persons with localized and disseminated disease. And you can see how it takes a number of weeks for persons with localized disease to catch up in their antibody level and detection rate in standard two-tiered serology based on their different clinical presentations. There are other factors involved in this difference, but I think this is one that's well documented in these studies and easily understood. strain differences, individual immune responses, things like that also play a factor, but this is a fairly easy one to grasp. So where are we in standard two-tiered serology? As we've seen before, there's clearly a need to improve detection in early disease. Under 50% is not really an acceptable level to utilize laboratory or to depend on laboratory diagnostics with a high degree. And a later disease performance actually does quite well. So back to this previous slide, what are our needs as far as diagnostic serology is concerned? An increased sensitivity in early disease is an important thing that we're working on. We do want to maintain the specificity, and I only alluded to this briefly. The specificity is achieved by two-tiered serology, and there is recognizably, particularly any of you that are doing these laboratory diagnostics or clinicians that are trying to read them, there's a need to simplify the complexity, particularly that associated with Western blots. There have been a number of improvements of Western blotting over the past years. I won't have time to get into that. I'm going to move to, in the next couple of slides, some modifications to two-tier serology that have been published in the last few years. And I will just briefly mention data associated with one of those modifications that utilizes VLSE or C6 antigen as part of a two-tiered approach. As I mentioned, we would very much like to reduce the complexity associated with Western blots, but maintain the specificity. So all these things are kind of summarized in this, the question at the bottom. Can parts of standard two-tier serology be switched or modified to obtain or achieve these objectives? This is a piece of a study that was published a few years ago and has been reproduced more recently, on references for those reproductions to be shown in the subsequent slide. But I'd like to focus your attention really just on two parts of this slide: the standard two-tiered algorithm performance and what I'm going to call a modified two-tiered assay, which is essentially two ELISAs. As I mentioned before, the second one in this particular piece of data is a C6 assay, and looking at this performance in these two assays in all stages of disease. And then while I'm here, I won't address this again later. One can see that the specificity in these two approaches, the standard two-tier testing and a modified two-tier testing, is essentially equivalent and at a level of about 99.5%. If you look at early disease in these two comparative algorithms, you can see a significant increase in detection of early disease, going from 42 to 53%, and that continues into stage 2 illness, jumping from 73% to roughly 100%. And a summary of that is depicted, although I would have to admit, looking at it now, rather poorly in my graphics in this inset on the right. But the bottom blue line is standard two-tiered serology, and the upper line is the modified 2-EIA approach. So in summary, where are we? Standard two-tier testing has been recommended for now 20 years. Its performance, particularly in early disease, is correlated with dissemination and even a matter of days, duration of illness. There's still a recommendation that all serology tests should go through FDA clearance. Then looking forward, what can we expect in the future? I believe that modified two-tier testing, I've only presented a few examples of that, but it affords similar or improved performance. It greatly simplifies testing, makes possible that both tests could be done in most laboratories, much more objective. Western blotting, I would not suggesting that Western blotting will go away, and there are specific applications and utilities of that, but I won't get into that here today. All tests, modified two-tier testing, other testing algorithms which we really haven't gone into in much detail, but might include PCR or culture, and novel technologies, which we'll hear some about in the next presentation by Dr. Mullins. All of these tests need to be rigorously validated. Comparative testing with or against standard two-tier testing is really the only way to realize performance improvements. And multi-site studies are also important in all these new or modified recommendations. With that, I'll stop. I think, Dr. Mead, that we're going to take questions at the end, but I'll leave that up to you.
Thank you, Marty. Yes, we'll save the questions for the end. Certainly, viewers can type in their questions as they occur to them, but we'll ask them at the end. I think we'd like to move on to the next speaker. Dr. Mullins, are you there?
Yes, thank you, Dr. Mead. And hello to all of the participants. It is really a pleasure to be doing this webinar today and to share with you some of our recent findings on the possible role of metabolomics in Lyme disease testing. For my talk today, I will give a brief background on Lyme disease, reiterate some of the aspects on diagnosis of Lyme disease, and describe our study approach using metabolomics. I will also share brief highlights from 3 studies. These 3 studies are actually being performed as collaborations between investigators at Colorado State University, New York Medical College, and CDC. I will then describe future directions and end with acknowledgments. Lyme disease is a serious problem in the United States, and as Marty mentioned and Paul mentioned, there's an estimated 300,000 cases occurring each year in the United States. The main causative agent of this infection is Borrelia burgdorferi. A picture of this bacterium is shown in the top right. Lyme disease occurs in humans through the bite of an infected Ixodes tick. The 2 ticks known to transmit Lyme disease in the United States are shown in the 2 pictures here. Lyme disease is also the most commonly reported vector-borne illness in the United States, and from the map shown, you can see that cases are heavily concentrated in the Northeast and in the upper Midwest regions of the United States. For this talk, it will become important to keep in mind that other pathogens are also transmitted by the same tick that causes Lyme disease. These pathogens can cause human granulocytic anaplasmosis, babesiosis, diseases caused by other Borrelia species, and Powassan disease. Lyme disease can be separated into 3 stages: early, early disseminated, and late. The majority of patients with early Lyme disease present with an erythema migrans skin lesion that can look like the one shown in the picture. Accompanying nonspecific symptoms during the stage can include fever, chills, headache, fatigue, muscle and joint aches, as well as swollen lymph nodes. Early disseminated disease occurs when the infecting bacterium travels to other parts of the body, causing the presence of multiple erythema migrans skin lesions, cardiac and/or neurological complications. Late Lyme disease in the United States usually presents as arthritis. As Marty mentioned, the recommended laboratory testing for Lyme disease is a 2-tiered approach. The first tier is usually an enzyme immunoassay that, if positive or equivocal, is followed by Western immunoblotting. If Western immunoblotting is positive, then this results in a positive Lyme disease diagnosis. The sensitivity of 2-tiered testing increases as the infection disseminates, but is poor in early stages of infection. For this talk, early Lyme disease is defined as having a duration of illness of up to 3 weeks. Of note, there are an estimated 3.4 million diagnostic tests performed annually for Lyme disease. The goal of our studies was to see if we could develop a usable, non-antibody-based laboratory test for early Lyme disease. To do this, the test must achieve similar or higher sensitivity than 2-tiered testing, and it needs to retain high specificity. The sensitivity for two-tiered testing, also as Marty mentioned, is 29 to 40%, and specificity is typically above or equal to 95%. It should be noted that specificity is dependent on the type of control samples used. Any developed test should be practical in the setting of a clinical laboratory, should be non-subjective, and ideally should differentiate between previous and active infection. We decided to try a metabolomics approach, which has only been used sparingly for studying infectious diseases. What is metabolomics? Well, metabolomics is the characterization and identification of small biomolecules, also known as metabolites, that result from cellular processes. So we're really looking at cellular end products, and these are usually small, typically less than 1,500 Daltons. The types of small molecules that we target through this approach are sugars, small peptides, lipids, amino acids, fatty acids, and nucleotides. Included are almost all molecules except for proteins, DNA, and RNA. The first step to our workflow is to process small volumes of the sample type being tested. We currently use serum or urine. All the studies that I will share with you today have been performed using serum. Processing of the sample removes all the proteins and large molecules. The metabolites that are left are then analyzed using liquid chromatography mass spectrometry, also known as LC-MS. A spectra of metabolites is generated for each sample tested. These are then combined into the groups being compared. For example, we can compare patients with early Lyme disease and healthy controls. Extensive computational analyses are then performed using specialized software, and this results in a list of differentiating metabolites, and this list actually provides their predicted chemical formulas and structures. The list of metabolites can then be used for biochemical pathway analyses or to develop disease-specific metabolic biosignatures. This talk will focus on the development and use of disease-specific metabolic biosignatures. So let me show you some of the results we have so far that look promising. Our first study, where we used the metabolomics approach to detect early Lyme disease, was a proof of concept study. We developed a metabolomic biosignature consisting of 95 metabolites to differentiate early Lyme disease patients from healthy controls. Classification modeling refined this biosignature to 44 metabolites and resulted in an average sensitivity of 88% and average specificity of 95%. If you remember from my previous slides, the sensitivity of two-tier testing is 29 to 40%. Additionally, patients with other diseases that can be cross-reactive in serology testing and that were not used in the biosignature development or model training were tested. A specificity of 93% was observed for these diseases. Of importance, in this study, we were able to correctly classify as positive up to 95% of two-tiered negative confirmed Lyme disease samples. Following our successful proof of concept work, we optimized our protocol and developed a new early Lyme disease versus healthy control serum biosignature. This metabolic biosignature consisted of 544 metabolites. This was refined to 68 metabolites with lasso classification modeling. This is a process that also performs feature selection. Testing of the classification model with samples that were not used in biosignature development or the training of the lasso classification model achieved a 92% accuracy for acute early Lyme disease and 96% accuracy for healthy controls. This is shown here as I pointed. Additionally, we used early convalescent Lyme disease samples to test this lasso model, and we were able to achieve an accuracy that was similar to that of early acute Lyme disease samples. The two-tiered testing results for the three groups tested are shown in this column here. The specificity obtained using the metabolic biosignature is slightly lower than that of two-tiered testing. but is still within the commonly observed specificity range for Lyme disease testing. The sensitivity of the metabolic biosignature attained, however, is much higher than that of two-tiered testing for both acute and convalescent early Lyme disease. Although it is important to understand the performance of healthy controls when you're developing a novel laboratory diagnostic test, healthy controls are really not what is tested in a clinical setting. Therefore, the challenge for us was really to evaluate whether our metabolomics approach could be used to differentiate a condition that is clinically similar to early Lyme disease. Shown here are some of the differential diagnoses for early Lyme disease when an erythema migrans skin lesion is present. The photos depict skin lesions and rashes that can be confused with an erythema migrans rash of Lyme disease. This includes an illness known as STAR-I, which stands for Southern Tick-Associated Rash Illness. This illness is associated with the bites of an Amblyomma americanum tick, also known as the Lone Star tick, which is different from the ticks involved in Lyme disease infection. Patients with STAR-I present with a skin lesion that can be indistinguishable from the erythema migrans rash of Lyme disease. The most frequent symptoms of STAR-I are nonspecific and overlap with those of early Lyme disease. And there is currently no known etiologic agent for STAR-I. Importantly, there is no available laboratory diagnostic test for this illness, and STAR-I and Lyme disease occur in geographic regions that overlap. And for all these reasons, we performed our second study that I'll share with you now. For this second study, we developed a metabolic biosignature to differentiate early Lyme disease from STAR-I. This biosignature consisted of 261 metabolites. Using classification modeling again, both random forest and lasso, we were able to differentiate early Lyme disease from STAR-I with high accuracies. This figure here shows an example of how the two groups performed. Specifically, lasso classification modeling differentiated these two diseases with accuracies of 89 and 98%, and random forest had similar results, shown here. We then used the same biosignature to see how healthy controls would perform. Random forest and lasso were again trained with all three groups, early Lyme disease, STAR-I, and healthy controls, and separate sample sets were used to blindly test the models. The figure here shows an example of how these three groups performed. Early Lyme disease and STAR-I had similar accuracies as those that we observed in our previous model, and healthy controls performed well with resulting accuracies of 93 and 95%. The previous studies demonstrated that we could use a metabolomics approach and apply this to accurate classification of multiple patient groups. Thus, we performed a third study where we returned to our early Lyme disease versus healthy control biosignature of 544 metabolites and applied it using lasso classification modeling again to differentiate early acute and convalescent Lyme disease, shown here, from a broader number of tick-borne diseases, shown here, as well as healthy controls. As you can see in the column of percent accuracy, the modeling with 61 metabolites resulted in classification accuracies of 80 and 82% for our early Lyme groups, and anywhere from 80 to 90% for our other tick-borne diseases. These other tick-borne diseases included STAR-I, again, as well as human granulocytic anaplasmosis and babesiosis. We also achieved a classification accuracy of 98% for healthy controls, and this is highlighted here. When we compare our results to two-tier testing, shown in this column, our metabolomics approach achieved a much higher sensitivity for the early Lyme disease samples, and there's a slight drop in specificity for STAR-I, human granulocytic anaplasmosis, but a higher specificity for babesiosis. Healthy controls do very well in our model, although slightly lower than two-tier testing. It is important to note that all of these groups are being compared based on a metabolic biosignature that was developed for the differentiation of early Lyme disease versus healthy controls. So really the next step for us is to develop specific biosignatures for each of these control groups against early Lyme disease and combine these in order to pick the best differentiating metabolites. This should greatly improve the sensitivity and specificity of our assay. And now I'll talk to you a little bit about future directions. Although we're very excited about our results, I do need to introduce you to the diagnostic valley of death. And really what we're working on now is with our collaborators to move this test from a research finding to a functional assay. And this really is not an easy process. And there are many aspects that need to be considered for the development of a functional assay and to avoid this diagnostic valley of death. These include understanding sample control and stability. We are working on incorporating internal standards and applying the most robust normalization techniques to the data. We need to take into consideration the heterogeneity present among patients with Lyme disease. This will include evaluation of Lyme carditis, Lyme neuroborreliosis, and Lyme arthritis, as well as individuals treated for Lyme disease. We need to understand the performance of all age groups at risk. This is particularly important in children. And we are working to refine our biosignatures to better differentiate other clinically relevant diseases, similar to how we address STAR-I. And last, and I apologize that this isn't showing up so well, we are evaluating various LC-MS platforms and defining the most appropriate algorithms for optimal performance and use in a clinical setting. I would like to acknowledge and thank the numerous investigators who have worked on these projects. As I mentioned earlier, the work presented is a collaboration between Colorado State University, New York Medical College, and CDC. We have also received resources from investigators at the University of Central Florida and Tufts University, and the funding sources for this work include CDC and NIH. Thank you.
Great. Thank you very much, Dr. Mullins. Our next speaker will be Dr. Cecilia Cato.
Hello. Good afternoon. Thank you for the opportunity for me to talk to you today about rickettsial disease diagnostics. I'll provide a little background, our current testing, as well as enhancements to diagnostics that we're currently working on. But first a little background. Rickettsial diseases in the United States include spotted fever rickettsiosis, typhus group rickettsiosis, ehrlichiosis, and anaplasmosis, most of which are transmitted to humans by ticks, although some of which are transmitted by other arthropods, including mites, ticks, and lice. Anaplasmosis, ehrlichiosis, and spotted fever group rickettsiosis in the U.S. are nationally notifiable diseases, and case reports indicate that they have been increasing since 2000. Common initial symptoms of these tick-borne diseases include fever, myalgia, headache, and malaise with or without rash. Fatality rates are highest in Rocky Mountain spotted fever patients at 5 to 10%, 3% in those with ehrlichiosis, and less than 1% in patients with anaplasmosis. Tests available at CDC include serological testing by indirect immunofluorescence antibody assay, immunohistochemistry, and molecular detection by PCR. Cell culture is used for isolation of microbiological assessment and not commonly used for diagnosis. IFAs have been the gold standard for rickettsial disease confirmation. Confirmatory testing can only be performed post-convalescence and with simultaneous testing of paired acute and convalescent samples. The patient must have clinically compatible symptoms, and endpoint titers must show a 4-fold or greater increase between the two samples. Probable cases are those with elevated titers for at least one of the samples and a compatible clinical history. If the patient has elevated titers for a single sample without a compatible clinical history, then the case is described as a previous exposure. IFA testing is the indirect assessment of patient serum for the presence of antibodies when the serum is exposed to agents of rickettsial disease. A convalescent sample is needed because antibodies are not typically present at the acute stage of illness. And an acute sample is also needed for comparison in order to see a rise in titer to determine a recent infection. Assay limitations include low specificity within genera and as well as subjective analysis. Because of the late presentation of reliable antibody titers, serology can only be used to confirm a diagnosis after recovery. Acute stage testing is mainly performed by molecular detection by PCR in tissue, blood, and serum, and by immunohistochemistry in tissues. Samples for culture should be drawn before doxycycline treatment, while samples for PCR and IHC must be taken before or within 48 hours of doxycycline. Immunohistochemistry testing is performed at the Infectious Disease Pathology branch at CDC. This process is for the direct detection of agent in tissue sections. Benefits include high specificity and the ability to identify other causative agents. Appropriate samples include fresh or formalin-fixed biopsy specimens taken before or within 48 hours of doxycycline treatment. These tests are limited to a few reference laboratories. Cell culture is performed for obtaining an isolate from patient sample for microbiological confirmation and is not commonly used for diagnosis because of the difficulty in this technique and the long period of time before growth is observed and verified. These tests are preferably performed with samples that have been taken before doxycycline treatment and include fresh blood and tissue biopsies. For ehrlichiosis and anaplasmosis, blood smears can also be performed because of the localization of the agents of these diseases within monocytes and granulocytes, respectively. However, it's not recommended because of the lack of sensitivity of this technique. Rickettsia have notably low levels of bacteria in peripheral blood because they are obligate intracellular organisms. So blood smears are not appropriate tests. So from the bite of an infected tick, bacteria may enter the endothelial cells, and the organism spreads cell to cell in tissues. Rickettsia then may enter the blood and circulate through the body. Since 2012, we've developed real-time PCR assays for Rickettsia species, Rickettsia rickettsii, Ehrlichia species, Ehrlichia chaffiensis, Anaplasma phagocytophilum, as well as Rickettsia prowazekii, Orientia tsutsugamushi, and Coxiella burnetii. The assays are used in our CLIA-compliant laboratory and have been shown to be sensitive and specific with a limit of detection of about 10 copies per reaction. These real-time PCR assays are rapid and effective methods for detection of DNA during the acute phase of illness. For molecular detection, the timing of sample collection is critical. Blood, serum, eschar swabs, tissue biopsies, and formalin-fixed tissue samples from the acute stage of illness can be tested. Sensitivity often decreases after 7 days from collection. Once again, samples must be taken before or within 48 hours of doxycycline treatment. And outside of the CDC, these tests are limited to a few reference laboratories. Along with the clinical support we provide to public health labs, we have also been working to enhance the availability of acute rickettsial diagnostics. So we have been working with the Laboratory Preparedness Response Branch on rickettsial real-time PCR assays. These assays are currently undergoing pre-market review by FDA. This will increase the national capacity for testing of these diseases at the state level. We are also trying to enrich for rickettsial targets by attempting cell culture amplification prior to PCR. By optimizing the cell culture process, including volume, specimen processing, and culture sampling methods, we're hoping to sample this culture within 3 days of inoculation for increased target numbers that may increase detection in samples with low bacteremia. We're also working on the development of new real-time PCR assays that target rickettsial RNA in order to increase the sensitivity of our molecular assays. Preliminary results show high analytical specificity, increased sensitivity by at least 100 times the current assay's limit of detection when testing non-fatal and fatal case patients. Further validation, though, is currently in progress. We're also currently applying this approach to other rickettsial diseases and Q fever diagnostics in order to increase the sensitivity of all of our assays. In summary, treatment of rickettsial diseases should begin upon suspicion of the disease based on a compatible clinical history and symptoms. Laboratory diagnosis is therefore for confirmation. Acute samples should be collected before or within 48 hours of treatment. Samples include blood, serum, swab of unroofed eschars, or tissue biopsies. Serological testing is for the confirmation after the resolution of illness, but should include both acute and convalescent sera in order to confirm a recent infection. So, thank you very much for your time, and I'll be happy to answer any questions. Thank you.
Thank you, Dr. Cato. Again, we'll hold questions for the end. The next speaker is Dr. Elias, but I'm having difficulty bringing up her slide. Very good. Thank you, Dr. Elias.
Good afternoon, everyone. My name is Maliha Elias, and I'm the Program Officer managing the Lyme Disease Research Portfolio at the National Institute of Allergy and Infectious Diseases. I'll be giving you a brief update on the Lyme Disease Diagnostics Research Portfolio at my institute today. This slide gives an outline of my talk today. I'll start by discussing the NIID Lyme Disease Research Portfolio and what it covers, followed by a slightly closer look at specific applications and topics within the portfolio.
As part of that discussion, I'll also give a brief update on the awardees of our targeted initiative on Lyme disease diagnostics that was released in fiscal year 2011. I'll also briefly touch on some of the recent publications on Lyme disease diagnostics that have resulted from work funded by NIID. I'll end the talk by discussing the new initiatives for Lyme disease. In terms of the framework of the U.S. Department of Health and Human Services, National Institutes of Health focus on conducting and funding basic and applied research, as well as developing medical interventions and developing research infrastructure. The Lyme disease research portfolio is housed within the National Institute of Allergy and Infectious Diseases, which is one of 27 institutes and centers making up the National Institutes of Health. Very quickly, the mission of NIID is to conduct and support basic and applied research to better understand, treat, and ultimately prevent infectious, immunologic, and allergic diseases. So as such, NIID has a dual mandate in that this institute is charged with maintaining and growing a robust basic translational and clinical research portfolio in microbiology, infectious diseases, immunology, and immune-mediated diseases. We're also at the forefront of responding to emerging and re-emerging threats to health through timely and appropriate research efforts. NIID intramural engages in research while the extramural side funds research in basic and applied sciences. The extramural Lyme disease research portfolio covers topics in basic research such as vector biology and pathogenesis, as well as translating basic research into products such as vaccines and diagnostics. I'm sorry that some of this is not very easy to read. It also covers clinical projects such as trials on xenodiagnosis and assessing persistence of infection after treatment. NIID also provides resources for Lyme researchers. That little yellow box at the bottom. One of those are the CDC NIH Lyme Sample Repository, and as well as our preclinical services. So our preclinical services are essentially contract-based services that cover everything from efficacy testing to toxicity and even manufacturing. And they're a great resource for product developers working on drugs, vaccines, and diagnostics. More information on those are available on our website. The NIID extramural program focuses on basic biology and pathogenesis of Borrelia species, such as the development of Borrelia-specific genetic tools and looking at new protein targets for diagnostics. This portfolio, as I mentioned, also covers applied research for diagnostics and vaccines. Diagnostics are a major focus of our portfolio, given the need for a sensitive, specific, rapid point-of-care diagnostic for not just determining early infection and active infection, but also for assessing treatment response. NIID released a targeted funding opportunity announcement, or FOA, in 2011 for Lyme disease diagnostics, which resulted in 4 made for exploratory or developmental grants, a mechanism that we call an R21, which covers up to 2 years of funding. The Institute recently also released 2 program announcements that cover novel approaches in treating, preventing, and diagnosing Lyme disease and other tick-borne co-infections. I'll talk a little bit more of that later. Here's a quick snapshot of the Lyme disease research portfolio before I start talking specifically about diagnostics. As you can see, it covers various topics such as tick biology, genetics, genomics, and host response. Although a large chunk of the portfolio focuses on pathogenesis, approximately 1/5 of the grants in this portfolio currently cover diagnostics research. But that proportion varies from fiscal year to fiscal year. I just want to point out that all the information presented about ongoing projects supported by NIID in this webinar is from this public source of information, the NIH Reporter. If you ever want to search on what NIID has awarded in various research topics, NIH Reporter is the place to go, and the website has been given here. I also want to mention the NIID report on current efforts in Lyme disease research, which was first released in 2015 and summarizes the developments in major research areas related to Lyme disease, including pathogenesis, vaccines and prevention, as well as diagnostics. This report is now in the process of being updated for developments in research that have taken place since 2015 and will be available on the NIID Lyme disease website soon. So as I mentioned earlier, in 2011, NIID put out an FOA, funding opportunity announcement, focusing on improved diagnostic capabilities for select biodefense and emerging pathogens. Specifically, projects were encouraged to focus on approaches that provide substantial and significant advances over conventional serology-based diagnostics, using antibodies, considering host responses, and having the capacity to detect and differentiate multiple Borrelia burgdorferi strains. Projects that focus on the development of methods to discriminate between active and previous infection and
focused on methods to identify stage of disease were also encouraged. So this FOA resulted in 4 awards, which focused on, among other things, multiplex platforms for diagnosing Lyme disease and assessing treatment response, the use of metabolic biomarkers and biosignatures in improving Lyme disease diagnostics, immune diagnostics, as well as cytokine basing and assays for Lyme disease. Most of the awardees utilized the CDC NIH Lyme repository, and 3 of these awards went on to the R33 phase, which basically gave them additional funding to continue and time to continue work on their projects. 2 projects funded through this FOA have also published on their work, and I'll mention those later in my talk. So this is a list of a few of the currently active Lyme disease diagnostics projects. Again, information on these projects is available through NIH Reporter, and this list also includes 3 of the awardees of the 2011 Lyme Diagnostics FOA. I'll start by talking about those. The second project on the list uses metabolic biosignatures and biomarkers to detect active Lyme disease, as well as effectively assess treatment response. Essentially, biomarkers, which are non-antibody molecules that arise in response to infection. The researchers are trying to see if a subset could be identified that is specific for Lyme disease. Something happening here? The slide? Oh, there we go. Okay, sorry about that. The slide kind of disappeared on me for a second. So the team working on this project has published on this work in 2015, and I have included that reference in a later slide. And the publication basically reports on a group of biomarkers that can differentiate between Lyme and a group of other diseases with high sensitivity, and that could accurately indicate Lyme disease in many blood samples that have tested negative through the currently accepted 2-stage antibody test. Claudia spoke a little bit about that project. Another one of our awardees is working on a cytokine-based immunoassay for Lyme disease, which utilizes a panel of Borrelia burgdorferi antigens in skin, and which are required for Borrelia burgdorferi infection and persistence in mammals. The development of this cytokine-based immunoassay, if successful, could allow for earlier and more rapid diagnosis of Lyme disease. We also have an awardee, Monica Embers, on this list. Her team is developing a multiplex technology which combines 5 Borrelia burgdorferi antigens into a fluorescent cytometric bead-based assay to detect Borrelia burgdorferi antigenic-specific IgD antibodies. Other projects on this list are also identifying biomarkers to develop prototype immunoassays using biomarker discovery platforms. They're developing urine-based tests using Borrelia burgdorferi protein biomarkers to detect early Lyme borreliosis, and developing antigen-based serologic assays that can complement the current serologic tests and potentially allow clinicians to objectively base decisions on additional courses of antibiotics, on whether to use additional courses of antibiotics to treat symptoms due to incompletely treated new infections. Several of our investigators are also working on the development of a new rapid point-of-care Lyme diagnostic using lateral flow technology. This slide captures some of the recent publications, which are a result of NIAID-funded projects on Lyme disease diagnostics. 2 of these publications, the Embers et al. Paper and the Mullins et al. Paper, are a result of the work performed under our R21, R33s that were awarded through our Lyme disease diagnostics FOA. The other studies highlighted through these publications focus on microfluidics-based point-of-care diagnostics, epitope-based serodiagnostics for Borrelia burgdorferi, as well as real-time multiplex PCR assays to detect the presence of Borrelia burgdorferi, as well as other pathogens such as Babesia and Anaplasma. Again, all of this is available through NIH Reporter. So NIAID has recently released 2 program announcements for novel approaches to understanding, preventing, and treating Lyme disease and tick-borne co-infection. Applications under these program announcements are encouraged to focus on gaps in our understanding of Borrelia burgdorferi pencilado infections, new Lyme-related borrelioses, and Lyme co-infections, with the goal of advancing the field towards solutions for better detection, prevention, and treatment of Lyme disease. These program announcements mainly differ, as you can see, there's 2 links there. They differ in terms of the mechanisms they cover. The R01 mechanism covers up to 5 years of funding, while the R21s are for more exploratory work and usually go up to 2 years. These program announcements became active in September 2016, and they will remain active till September 2019.
The pay lines, due dates, and study sections for these program announcements are the same as for any other investigator-initiated application. And since these announcements have not been active for a year, we do not yet know whether they've resulted in an uptick in Lyme disease-focused applications coming in and being funded, but we hope that that will be the case. So more information on these is available on this website. I'd like to end my talk by highlighting the fact that Lyme disease diagnostics research is a high priority for NIAID, given the need for diagnostics which detects early and active infection, that can assess response to therapy, and which can distinguish across multiple Borrelia strains. NIAID will continue to support high-quality science in the form of research project applications for Lyme disease diagnostics research, especially supporting further development of successful early-phase projects. We hope to continue facilitating continued collaboration with our CDC colleagues, and we encourage principal investigators to contact FDA when validation of a proof of principle research project is underway. NIAID will continue to fund research in other areas within NIAID Lyme disease research program. Some of those are mentioned in this list here. For example, new and emerging Borrelia species like Borrelia miyamotoi and Borrelia meionii. Thank you for your attention, and I can take questions at the end of the talk, at the end of all the talks.
Great. Thank you very much for that. I believe we'll be loading up the next slides for our final speaker, Dr. Gerald from FDA.
Okay, great. Thank you. My name is Noel Gerald, and I'm a biologist and scientific reviewer at FDA. And I work in the group that reviews infectious disease diagnostics for most of the infectious diseases, and definitely including all of the tick-borne diseases that we've been discussing so far. So in my talk, I'm going to give a brief overview of what we do in our group. This will be familiar to those who've heard me speak before or who may have seen the earlier webinar. And then I'll talk a little bit about our evaluation of pre-market applications for Lyme disease IVDs for the serology. And then I'll wind up with just a plea for people to use the FDA pre-submission program to interact with us early when they're considering sending a pre-market application. And then I'll finally just give a wrap-up of all of the previous talks. So just starting with the regulatory definition of in vitro diagnostic devices, which would be reagents, instruments, and systems intended for use in a diagnosis of disease in order to cure, mitigate, treat, or prevent disease, and also the devices for the collection, preparation, and examination of specimens from the human body. So FDA has pre-market review and post-market reporting, and this is what ensures the tests on the market produce accurate and reliable results so that physicians can make informed decisions to best treat their patients. One clarification I often have to make for people, the FDA is not conducting the tests themselves. In pre-market review, we are reviewing the evidence of, that supports the claims for a device that someone submits to us in a pre-market review application. And so that's mainly what I'm going to be talking about in the rest of my talk. So here are a few principles of a successful IVD pre-market submission to FDA. And still, first and foremost, is to have a clear and precise intended use. The intended use is the driving force of our scientific review. And what I mean by clear and precise is to outline who will be tested, where and when, what are the appropriate specimens, and more importantly, how are these results going to be used in patient management? Are we talking about a first-tier test? Are we talking about a second-tier test? Is it a standalone test? These all go into weighing the risks and benefits of your device. We, of course, want a complete device description in your application, and then there will, of course, be evidence that should support your claims for the intended use. You should have a good manufacturing quality system in place. And it's important that you have labeling that includes all of the instructions, limitations, and a summary of performance characteristics that an end user would need to know. So moving to the example of serological tests for Lyme disease, we do have a guidance document for these types of tests, and you can find it at the link at the bottom of the slide. It was published in 2013, and basically this provides the outline of all the recommended clinical and analytical studies that we expect to see in a pre-market review. It's applicable to Lyme disease serology IVDs that fit into the two-tier algorithm, the current algorithm that Marty described, either as a first-tier test EIA or a second-tier test Immunoblot or Western blot. But it will have, even if it's not one of these, it's still a good starting point because many of the concepts will apply to other disease types. So when you go and you look at that guidance document, it will talk about the clinical and analytical studies. You can look there in detail. I'll just mention a few highlights from there. For clinical studies, there will be testing samples from your intended use population, patients suspected of Lyme disease. And it will also include testing with well-characterized samples from confirmed Lyme disease cases at different stages of disease so that we can report on the different stage-specific performances that both Marty and Claudia discussed earlier. And the clinical studies will also include testing with CDC reference panels, samples from endemic and non-endemic areas. You can see the guidance. And then under analytical studies, again, as alluded to by Claudia and Marty, we look at reproducibility. We look at cross-reactivity, how likely is your assay to be false positive with specimens from another disease. And then the effects of the different types of samples that you're testing and any interferences that may be possible that an end user should know about when using your test. So while you can find a lot of good guidance that's overall in general in the guidance document, if you would like more specific advice that's particular to your certain platform or assay, this is where we would highly recommend that you take advantage of the pre-submission process. So pre-sub, as we call it, is a non-binding and confidential process for obtaining FDA feedback. So this can be submitted at any time during product development. Early is good. And it can be used to discuss many types of questions, including the regulatory pathway, questions about clinical and analytical study design, the scientific evidence and performance data that's used to support your intended use claims, questions about the appropriate reference and comparator methods. So this, it need not be an all or nothing process. This is an iterative process, which means, if you just have a question about, say, regulatory pathway, you can submit that. And then later on, when you have a question about this particular study design or a particular reference method, you can send in another pre-sub. The details of submitting it and the ways you can get your feedback can all be found at the link at the bottom of the slide. And so fitting this into what we've heard previously, we've heard about different platforms for testing Lyme disease, for example, that have nothing to do with serology, or different ways to interpret some of the current serological tests that probably are on the market. And I just want to highlight again that these are the types of things that other device manufacturers have successfully brought to the pre-submission process. And we feel that it has really helped ease their transition to market once they send in their actual pre-market application. So here are some of the common questions which we have addressed from device manufacturers from a wide range of IVD platforms and diseases, not just tick-borne diseases, and conditions. So one example is how to establish the IVD performance for a new disease, how to introduce new technologies that may raise new questions of safety and effectiveness, or just in general, how to address some new approach that a sponsor may have to improving patient health. So the take-home message of my entire talk would be this, is that FDA is open to discussing new approaches to diagnostics, whether it's for the diagnosis of Lyme disease or other tick-borne disease or other infectious diseases in general, and we encourage pre-subs to discuss this, whether it's, just getting into the benefits and risks to patient health of your particular proposal, or to have, an in-depth dialogue of the appropriate study recommendations that will support a new paradigm that is ultimately aimed at improving patient access and diagnostic accuracy. So with that, I'll leave you this link to the FDA Medical Devices homepage, where you can find all of the guidance documents I've mentioned in my talk, as well as guidance documents for other specific diseases. You can find the link to the medical device database that Marty referred to, and other resources for consumers, healthcare providers, and industry. And so, to wrap up this webinar, we've heard updates on the state of diagnostics and treatment for several tick-borne diseases, including Lyme disease and rickettsiosis. And we've heard about HHS research and funding initiatives that are aimed at improving tick-borne disease diagnostics. With Dr. Mullins, we heard about the research into the metabolomics-based test to differentiate early Lyme disease from other illnesses, which would be a complement to a serological approach. We heard from Dr. Schrieffer with his overview of Lyme disease diagnostics as a whole and a survey of serological applications, and the discussion of these alternative approaches that are being considered, the alternatives to the standard two-tiered testing algorithm for Lyme disease serology that we've had since the late 90s. And then we heard from Dr. Cato with a review of the different rickettsial diseases that are emerging and have continued to be an issue in our country, and the testing and treatment recommendations for them. And also heard a little bit about the research that they're doing into improving the diagnostics for those diseases. And then finally, we heard from Dr. Ilias at NIH and NIAID on the current funding initiatives for basic translational and clinical research on Lyme disease and other tick-borne illnesses, which all range from basic biology and pathogenesis to new diagnostics and vaccines. And then finally, I gave a background on our pre-market review process and a plea for everyone to use the pre-sub process if they're considering any of these or other new applications that will be improving diagnostics for tick-borne diseases. Thank you.
Great. Thank you very much, Dr. Gerald. We have a number of questions that have come in. We have a few minutes to answer some of them. The number one question we've been receiving is where can people get copies of this presentation. And this presentation, like previous webinars, will be posted on the CDC Lyme disease webpage. So it's www.cdc.gov/Lyme disease. Just Lyme. Please allow 4 to 6 weeks, however. That's the time that's needed to get the transcript and prepare the document. But all of today's talks will be provided there. A few additional questions, and actually, Noel, the first one I think is just for you. You mentioned stressing the pre-sub process. How much is there a cost associated with this, and who would they contact to do that?
So currently there's no fee associated with the pre-sub process. So the general contacts are in the guidance document that I provided a link to. But if there is some specific question, people are free to send me an email, and if I'm not able to answer it, I can forward it to the appropriate person.
Great, thank you very much. The next question, I believe, is for Dr. Elias. There may be a couple here. One is, are there any current or planned clinical trials on antibiotic prophylaxis following tick bite?
We do have some stuff in the pipeline. It's so early on that I can't really talk about it, but we do have some researchers who are interested in looking at that. I'm sorry I can't give more information because it hasn't sort of hit prime time just yet.
Great. A follow-up question is, can graduate students apply for NIH funding?
There are different mechanisms that are available to graduate students depending on what phase they're in their career. So I would really recommend that you speak to the program officer or whatever research area you're focusing on. So if it's Lyme disease, you'd be talking to me, and I can help you navigate that.
Okay. Thank you very much. A question for Dr. Schrieffer, and my apologies, I'm having a little bit of difficulty pulling these up, but it's been noted that the sensitivity of serologic testing appears to be best when acute and convalescent samples are sort of tested. Can you comment on the effects of antibiotic treatment on that process? How does that affect the sensitivity of serologic testing?
Sure. In general, if it alludes to two of the points that I raised earlier, duration of illness prior to treatment and dissemination. If a person is treated very early, within a day or two of infectious tick bite, the antibody response is going to be very much muted. If they have several weeks of untreated infection, the antibody response will be much greater, and that carries on throughout the course of untreated illness. Once treated, there is usually a continuation of building, particularly in early disease, for a couple of weeks, but at some point, effective treatment will stop the continued evolution of the immune response. The question about acute convalescent serum sample testing, I'm not sure if I completely understand that. It can be very useful, and I think the question might come from the perspective of if a person has an acute sample test that it's negative and then they're treated, can we expect the convalescent sample to be positive? It goes both ways, but again, the longer duration and dissemination of infection prior to that treatment, the more likely the sample is going to be positive.
Great, thank you. Dr. Gerald, another question for you at NIH. Do I gather from your comments during your talk at FDA, I gather from your talk at your comments that FDA is now considering other scenarios for testing, including single-step procedures or other modalities. Is that correct?
Yes. So basically, anyone can submit a pre-sub with a proposal for other modalities. So just because we have all of the current cleared assays are two-step doesn't mean that we're not open to discussing the scientific issues. And so yes, the answer is yes.
Okay, a follow-up question to further clarify that. There's a question of whether or not the modified two-tier test is already approved by FDA and when it will be available. And well, go ahead if you want to answer that.
Okay. So when we talk about the algorithm of whether it's the standard two-tier algorithm or the modified two-tier algorithm, that actually is not a separate item that FDA is clearing and not clearing. That's how those assays are going to be used. So they may be already cleared assays that would be used in that algorithm. So I guess the difference is, so no, so the technical answer is no, FDA did not clear a modified two-tiered test. However, if someone wishes to send in an application with an intended use that specifies my test should be used in this algorithm, that's something that we can discuss. I know it's kind of a, seems like it's a splitting hairs distinction, but it is an important distinction. And if I haven't answered the question, please re-ask it.
Great, thank you. There are a number of questions for Dr. Mullins, and I apologize. Unfortunately, she had to run off to another presentation that she was double booked on. I can see some of the askers of these questions, and what I will do is try and get her to address your questions by email separately. So I'll pass those along to her. Again, our apologies that we don't have that available. There is a question to clarify, will the slide deck be downloadable before the 4-week required for transcribing the entire webinar? And the answer to that is no. I think we've really hit most of the main topics. I have one other question for NIH. Do the new program announcements allow applications to focus on other tick-borne infections, such as Babesia or Anaplasma alone, or are they intended only as part of co-infections for Lyme disease?
So yes, in short, they are allowable. As long as it's a possible Ixodes-transmitted co-infection, you can study it alone, such as Babesia and Anaplasma, without the need for the co-infectious states being part of the application. So in short, yes, those are allowed.
Okay, terrific. Thank you. And I would like to end, our time is up. I'd like to end by thanking all of our presenters today for their presentations. I think hopefully it's a large and complicated topic, but I think we've gained some new insights and from a variety of different avenues. So thank you all for your presentations, and thanks to everyone who tuned in for today's presentation. Again, the full presentation will be available in 4 to 6 weeks at www.cdc.gov/Lyme. Thank you very much. Have a good day, everyone.
That does conclude today's call. Thank you all for participating. You may disconnect your lines at this time.